Welcome everybody to today's webinar. The goal of the webinar is to use our laser printing technology and highlight the broad application range of the peptide microarrays by means of several case studies. PEPperPRINT is a spin-off of the German Cancer Research Center located in Heidelberg and has long-time research experience, including publications in high-impact journals like Science. Furthermore, in 2010, we received the Innovation Award of the German industry for our platform technology, which is broadly patent covered. The technology is based on a peptide laser printer, which allows direct synthesis of peptides on glass slides. An image of the laser printer can you see on the top right here on the slide. Instead of the 4 color cartridges which are present in a normal office laser printer, our printer contains 20 amino acid cartridges. You can see the cartridges here on the picture below. Each cartridge contains microparticles with 1 activated amino acid embedded in the toner material. After simultaneous printing of all amino acids on glass slides, all microparticles are melted. This is shown in the little cartoon here on the top. So this is the printing step and the next is the melting step. During melting, the activated amino acids are released, initiating coupling to the linker on the glass surface or the previous amino acid. The cycle of synthesis is completed when excessive monomers are washed away and the FMOC deprotection group is removed from the N-terminus. Then the next layer of amino acids is printed. The benefit of this technique are its unique digital printing flexibility in terms of peptide content, high spot density, as well as a significantly reduced material consumption, resulting in very attractive chip prices. In contrast to spotted arrays, which require a high upfront investment for peptide synthesis, we synthesize all components directly on the array in the exact quantities required for your experiments. This saves material, time, and cost. First, I would like to focus on the pepper chips, which are the peptide microarrays. The pepper chips can be customized for standard peptide arrays with up to 9,000 spots per chip in variable peptide length. We can print any target protein or peptide collection within 2 to 3 weeks. And depending on the size of the array, multiple array copies can be printed, which can be assayed individually. On the left, you can see a schematic of the different array layouts with different numbers of array copies per slide. The chips are mostly applied to identify specific amino acid sequences, which are called epitopes. They are bound by antibodies. Therefore, we developed a standard protocol which works for purified antibodies as well as antibodies from sera. And since our chips have standard slide format, they are compatible with most chip readers and can be easily handled and analyzed by you. And beside custom arrays, we also provide peptide arrays with standard peptide content for infectious disease, cancer, and autoimmune research, as well as biomarker discovery. Standard pepper chips for infectious disease research cover entire viral proteomes, for example, of the dengue virus, the hepatitis C virus, or influenza virus, or they can also cover selected viral antigens that are translated into overlapping peptides. On this slide, you can see a schematic how epitope mapping works. We just need the amino acid sequence of the antigen to which the antibodies bind. The sequence is then translated into overlapping peptide by the PepSlide Designer software. We routinely synthesize 50-mer peptides with a peptide-peptide shift of 1 amino acid, or which equals a maximum peptide-peptide overlap of 14 amino acids. The peptides are then directly synthesized on the glass slide. Their array is subsequently stained with a target antibody that binds to its specific epitope and a secondary antibody with a fluorescent label. An image of the array is taken, which you can see here, and this is done, of course, with a microarray scanner. And since we are using spot duplicates, each signal appears twice, so you can see 2 rows of signals here. After the data acquisition, the Peptide analyzer software immediately then calculates the common motif in the peptide hits. So the common motif is in this red angular here. And this motif is then the epitope that was bound by the antibody. On the left, you can see 2 examples of such an epitope mapping. One with a monoclonal sample binding to a single epitope, and polyclonal sample binding to multiple epitopes. And each array is surrounded by HA and flag peptides, control peptides, which are visible as red and green ribbon. You can see it here. In the first case study, the customer intended to compare the influence of different adjuvants on immunization. He immunized rabbits with the antigen lactalbumin alone, as well as covalently bound to adjuvant, which he called number 4 and number 8. We translated the antigen lactalbumin into overlapping peptides and printed 4 array copies per slide. We subsequently incubated the arrays with antibodies from the immunized rabbits. And immunization with lactalbumin alone, which you can see here, without any adjuvants, did not induce antibody binding to lactalbumin. Rabbits which were immunized with adjuvant number 4, it's depicted here, they developed antibodies that recognize 3 epitopes. These epitopes are here, the second is here, and the third is here. And in contrast, antibodies from rabbits that were immunized with adjuvant number 8 bound to 3 additional epitopes, and this means that adjuvant 8 increased the immune reaction against lactalbumin. So by use of the peptide microarrays, we could provide the customer with accurate molecular data about the immunization effect. The next example illustrates a time-dependent development of the humoral immune response in an immunoglobulin mutant mouse that was immunized with an HIV antigen. We translated the antigen into overlapping peptides and analyzed on one single chip the epitope pattern of the immune serum and of sera taken 2 weeks and 4 weeks after immunization. So no antigen-specific antibodies could be detected prior immunization, but 2 weeks post-immunization, one single epitope was discovered, which gave the fastest immune response and was obviously highly immunogenic. 4 weeks post-immunization, further epitopes could be detected, and 2 of them gave similar strong signals as the first one. You can also see it here, where the intensity is plotted over the peptide sequences. And here the blue line is the pre-immune serum, the purple line is the immune serum taken 2 weeks post-immunization, and the red line is 4 weeks post-immunization. And in case you are not only interested in one single antigen, but in the immune response to the whole pathogen, one of our standard microarrays may be of interest for you. The array in this case study here contained the entire dengue virus type 2 proteome translated into overlapping peptides. we stained the array with serum of a dengue virus patient, and we were able to detect two exceptionally strong epitopes here and here, which belong to the protease NS3 and the RNA polymerase NS5. In the following case study, we used the standard array for autoimmune disease. And this contains over 2,700 epitopes in duplicates, and they are linked to autoimmune diseases and are listed in the Immune Epitope database, which is an open source database in the internet. And among these peptides, around 180 epitopes that contain citrulline instead of arginine were printed on the array. Citrulline is a derivative of arginine and is often bound by antibodies in patients with rheumatoid arthritis. The aim of this study was the identification of autoimmune epitopes for diagnostic development. The client provided 4 serum samples from autoimmune patients that were hybridized to the Autoimmunity microarray. On the left, you can see 2 of the 4 arrays here. On the right, the signal intensity is again plotted over the peptides. All 4 samples bound to an epitope of a viral antigen. You can see the signals here, 1, 2, 3 signals. And when we looked for background information for this viral epitope in the Immune Epitope database, we found that the same epitope is also present in a human protein which is associated with diabetes. So the Autoimmunity microarray is an attractive tool to disclose autoimmune related antibody patterns in patient sera, for example. Another biomarker study was carried out using a custom peptide array to analyze the serum profile of glioblastoma patients. And here we compared long versus short time survivors with the aim to identify new prognostic markers. Each pepper-chip peptide array contained 6 tumor antigens, which were translated into overlapping peptides. 2 array copies were printed on each slide, and we tested then 25 serum samples and found 20 significant peptides. You can see them here. Of course, I cannot show you the peptide sequence since these are all customer studies. But what is really interesting is that the long-time survivors, the LTS, they recognize peptides differently to the STS, the short-time survivors. So we clearly found peptides that discriminate the 2 groups. So these peptides are now promising candidates for prognostic markers. Currently, we are testing 160 additional samples for hit validation. And in the future, a prognostic test on the basis of these peptide markers may not only help to stratify the patients, but also to select appropriate medication. In general, all arrays with standard slide format can be easily stained and analyzed on your own. The main prerequisite is a microarray scanner. And if you don't have the time or manpower or technical infrastructure to perform the assays, you may consider our PEPperMap service. We only require the antigen sequence with 10 microliter serum or 10 microgram antibody to run the assays in our labs. The service is performed by highly experienced staff and includes a custom peptide microarray with your antigen or antigens translated into overlapping peptides, all immunoassays with your antibody or serum sample and matching secondary antibody, of course, then control assays with anti-flag and anti-HA control antibodies, readout of microarray data, data quantification, evaluation, and at the end you get a detailed report with protocols, experimental details, and results. We offer 3 standard epitope mapping types along with our custom analysis service. The type 1 epitope mapping, which you can see on the left, includes one peptide or a peptide microarray with 1 peptide length and maximum peptide overlap. Here you can see the result of a monoclonal sample and a polyclonal sample. And when the epitopes are very close to each other, for example here, a type 2 mapping is helpful to distinguish them. The type 2 epitope mapping includes 3 different peptide lengths with their respective maximum peptide overlap. In the example here, we used 10, 12, and 15-mer peptides, and the long row of signals in the 15-mer analysis here could be traced back to 2 adjacent motifs in the 10-mer analysis, which you can see here. And if the epitope of an antibody or serum sample is already known, the permutation scan enables the determination of conserved and variable amino acid positions, as well as the identification of new epitope variants. An example of such a permutation scan is shown on the next slide. Here, each amino acid position of the starting peptide are replaced by the 19 remaining L amino acids. And since the printer has 4 additional printing units, we also incorporated D amino acids in the scan. In the example shown here, we scanned the HA epitope and incubated it with the anti-HA antibody. We printed 3 array copies with spot duplicates next to each other, which you can see here on the microarray scan. Beneath, the mean intensities for all peptide variants are displayed. So here is the peptide sequence, and on the top you have the different amino acids that were used to replace each position. So the tyrosine on the second position, for example here, this tyrosine is very conserved, since replacement of this tyrosine by any other amino acid reduces the signal. Only if the tyrosine is used, you get a high signal intensity. In contrast to that, the last 3 amino acids can be replaced by any other amino acids, suggesting that they play a minor role in epitope binding. You can see this here. And on the right, you have another description of the same results. And what is really interesting is that the tyrosine on position 7, which we were already talking about here, this tyrosine could also be replaced by D-glutamic acid, which is here the E in the schematic here. So the incorporation of D amino acids into peptides can help to stabilize the peptides by preventing them from protease digestion. So this can be then very interesting, for example, if you are working in peptide vaccine development. And beside the standard slide format covering 9,000 spots, we also provide large-scale peptide arrays which cover up to 52,000 spots, or approximately 26,000 different peptides and duplicates. The so-called Pepper screens are especially attractive for antigen and biomarker discovery, as well as peptide engineering. And since these large arrays are not compatible with all microarray scanners, we provide them along with our comprehensive analysis service. In this case study here, a large peptide array was used to discover new disease-relevant antigens in a two-step process. The first array, which you can see here, contained 334 selected antigens, which were translated into approximately 26,000 overlapping peptides in duplicates. So the array was first incubated with pooled control sera to detect background signals, which you can see here. And since these sera came from humans, a strong background is expected. Subsequently, the same array was stained with pooled patient sera, which you can see here on the right, and clearly revealed a number of antigens and epitopes, for example here or here, that were exclusively stained by the pooled patient sera. We selected then interesting antigens, antigen domains or epitopes, and printed them on an array with standard slide dimensions for further hit validation with high sample numbers and individual sera. And these kinds of studies are interesting, for example, if you would like to define immunogenic regions of pathogens for vaccine development, or if you would like to discover biomarkers from a large peptide dataset. And besides analyzing antibody binding, our arrays can also be used for peptide drug development. And in collaboration with the British company Isogenica, we were searching in a two-step approach for a peptide binder of TNF-alpha. In the first step, Isogenica applied its cis display technology, which is based on a library containing more than 10 to the power of 13 peptides. They performed several rounds of affinity-driven selection and ended up with 16,000 peptide hits. We then printed these 16,000 peptide hits on our large-scale arrays in triplicates. And we hybridized the array then with TNF-alpha. 4 significant consensus motifs could be identified, and in a permutation scan of the main lead motif, we could also then identify the amino acids that are most important for TNF-alpha binding. And currently, the peptides are being tested in vitro and in vivo assays. And here you can see a list of selected publications where you can find more details about customer projects, for example, and also about our printer technology. Taken together, our peptide microarrays are a powerful tool for immunological research, biomarker discovery, and drug development. And due to our proprietary laser printer technology, we can offer an outstanding content flexibility with up to 50,000 different peptides per array, accompanied by a fast production time and reduced chip costs. We follow high quality control guidelines to ensure chip integrity and reproducibility. In addition, it is very important for us to support the customer during project setup, answer critical questions, and discuss assay results to speed up your research. You may either contact us directly or via our representatives in the US and UK, or our distributors in the US, Japan, China, Korea. Finally, I would like to thank you for your attention, and I'm now available to answer questions.